was so volatile. A little color guidelines from the class. Yeah, to protect the flyway. I think about that too.
Take all the tests out there.
All right, so let's go ahead and get started here. I am slowly passing back those exam corrections. So if you haven't done those back, I will give you before you leave class today. Let's see, content check number 4 is due today, and we will go through the material today. What else do I have for you? I'm offering another study session tomorrow during the common meeting time. So Wednesday at 3:30. It's optional. Come if you want. Bring your questions. I will be there. I felt like with this shortened unit, I needed to offer yet another opportunity to come visit. I do have office hours Wednesday morning as well if you want to come visit me instead. Let's see, exam is on Thursday, so it is coming up. Yes, this material will be on the exam, but yes, I am taking into account that you will have just learned it today and will be tested on it on Thursday. So that being said, I can tell you that the bulk of the exam is going to be more in-depth and more focused on cellular respiration, energy and enzymes, fermentation, and anaerobic respiration. But there will be some questions about photosynthesis, right? But think more broad term. Let's see. All right, so I mentioned the content check. I'm actually giving you the question ahead of time, so as we go through lecture today, you can keep this in mind. We'll come back to this at the end of lecture. On your slides, this is actually at the end of the lecture slides, so you do have it, so you can write it down, take a picture if you need it right now. We'll be talking about this throughout class. Also, to facilitate just getting through the material and knowing that you will be tested on this on Thursday, I've already filled in all of the blanks on your lecture slides. You will notice that as you go through, all the red underlines are already filled in for you. Alrighty. Any questions before we get started? Great. So let's jump on in. We left off on this slide here where we talked about electrons capturing light energy. All right, so light comes in, photons come in, they excite the electrons within a molecule. Those electrons move into this excited state, right, this higher energy state. But this higher energy state is very unstable, right? So that energy needs to be quickly lost in order for this molecule to go back down to its ground state. Right? We talked about 3 different ways that this can happen. Right? Energy can be lost from this electron via heat, also via light. Right? So we talked about the bioluminescent animals emitting off light. That is a form of giving off energy. Plants also have the ability to give off light. In fact, pretty much all plants do this. It's just in such low quantities that we can't detect it with the human eye here, but they do fluoresce. But mostly what we're talking about today is the electrons going back to or giving off that excited energy by being transferred to another molecule. All right, so we're talking about excited electrons being transferred or captured. And that's how we're going to capture this energy from this excited electron. All right. So that's where we left off. Hopefully that rings a bell, because we're going to talk about this electron being captured. All right. So in a plant, the electron that is becoming excited from this light energy is an electron that's located within the pigments. And in plants, these pigments are most commonly chlorophyll pigments. So I have a diagram of a chlorophyll pigment up top here. Right? The electron is within this chlorophyll head. Right? We have this head and then we have this long tail. One of the electrons in this chlorophyll head is going to be the electron that gets excited from that burst of energy. All right, So again, when that electron gets excited, it goes to a higher energy state. It's carrying a lot of energy. It tends to be pretty unstable. Now, light is exciting this electron, but not all light is equal, right? Some light contains more energy than other wavelengths. So if we're thinking about the visible spectrum of light, right? We have greens and blues and oranges and reds, right? We have this whole spectrum. Each visible light that we see has a specific wavelength associated with it, right? So some wavelengths are really short, right? Really, really short wavelengths, while others are really long wavelengths, right? And it's the length of those wavelengths that determine how much energy that light is carrying. So chlorophyll pigments, right? Chlorophyll, they tend to absorb red and blue light. They do not absorb green. In fact, they reflect green, which is why we see plants as being green, right? Chlorophyll is reflecting that green back to us. But they do absorb the red and the blue. But the red and the blue have different energies associated with them. So if we look at this graph here, we can see we have different wavelengths, different photons, exciting an electron. We can see this blue wavelength, right, has a much quicker wavelength, much shorter wavelength to it than this red wavelength, which is much more of a long wavelength. So when blue light excites the electron, that electron gets really excited, right? Because that blue light contains a lot of energy. It goes to a pretty high energy state. Now red light can also excite this electron, right? But red light has a longer wavelength, so the amount that this electron is getting excited is not as much as when it was excited by the blue light, right? There's not as much energy associated, right? So, big picture, light is exciting the electrons. Depending on the wavelength, will determine how excited those electrons get. Right? Blue light exciting an electron more than, say, red light with a longer wavelength. And again, this is all happening in these chlorophyll pigments. That is where the electron is located that is becoming excited from this light. All right, so we're talking about chlorophyll pigments here. There's actually two different types of chlorophyll pigments in plants, two different main types, I should say. Chlorophyll A and chlorophyll B. I don't expect you to know the difference between the two, but I think it's interesting and that you should know that there's more than just one type of chlorophyll. The difference between chlorophyll A and chlorophyll B is just a little bit of a change in its molecular structure on that top corner. Right? And they do absorb slightly different wavelengths. All right, so we have our chlorophyll pigments. It's important to note that these pigments, they work together, right? One pigment does not do everything. Pigments actually work together in groups. All right, so chlorophyll molecules tend to group together. So in this photo here, we have the thylakoid membrane being shown. And within the thylakoid membrane, remember we're inside of the chloroplast inside of the thylakoid. Inside of that thylakoid membrane, we have this pocket of densely packed pigment molecules. Right? So they work in these groups. And these groups tend to be like groups of like a couple of hundred of pigments all together. And what happens is when one pigment, right, the electron in one pigment becomes excited. Remember how I said we have to get rid of that excited energy through heat, light, or transferring that electron? Well, here we're going to transfer that electron. Right, so if we're looking at this image in the middle of the screen here, all of these puzzle pieces are types of chlorophylls. Right, we have a photon coming in and exciting an electron on one of these chlorophylls. That electron is going to be transferred to another chlorophyll, which in turn will be transferred to yet another chlorophyll. And essentially just kind of bounces between chlorophyll pigments until it reaches the center of this complex, which we'll talk about in a minute here, until it reaches the reaction center where it can officially be captured. Right, so these pigments, they're working together. A photon comes in, excites an electron. That electron is transferring its energy by being moved from molecule to molecule, from pigment to pigment. And we have a name for this type of energy transfer. It is resonance energy transfer. Right? So the movement of that electron from pigment to pigment to pigment to pigment to pigment until it's finally captured, that is resonance energy transfer. And we're going to come back to this term a few times when we talk about the light reactions. All right, so pigments are working together to move this excited electron. But there are more than just chlorophyll pigments in a plant, right? Chlorophyll is by far the most dominant type of pigment, which is why we see plants as green, because we're mostly seeing the chlorophyll pigments reflecting the green back at us. But there are other pigments. They are there. They are just masked by the presence of chlorophyll. Right? We can have other pigments like these carotenoids. Right? If we look at the wavelength of light that they absorb, right, they tend to absorb more in this green spectrum. They have the ability to absorb some of this green light, whereas chlorophylls do not. So there are other pigments out there. I don't expect you to know the name of these other pigments, but I want you to know that they exist. And they exist to either absorb different wavelengths or to act in other means, such as sometimes they act like a sunscreen. They can protect the plant from excessive sunlight. Excessive sunlight can actually be very damaging to a plant. All right, so it's fall season. We see the leaves changing outside. Right, we're going from greens to some reds and yellows and oranges. Right? The reason we are seeing this change is because the chlorophyll pigments in these leaves are being broken down. And the chlorophyll are actually going back into the tree trunk, right? So that they can be recycled and used again in other means, maybe next year, maybe make new chlorophyll pigments. But they're being recycled, they're shoveled back into the tree trunk, and what's left behind are all of these other pigments that used to be masked by the chlorophyll. Right? So now we're starting to see some of these carotenoids or xanthophylls showing through. And these are the pigments that show up as oranges and reds and yellows. Right? So they're not going to go back into the tree. Right? They're staying in the leaves, they're staying put. But now the chlorophyll that was overly abundant before has now left, so now we can actually see these remaining pigments. All right, so pigments work together. Let's talk about the structure of how these pigments work together. Right, so if we have pigments that are grouped together in a thylakoid membrane, right, they have a name to this grouping, and that's called a photosystem. Right? So a photosystem consists of two parts. It consists of all of the pigments, and then consists of the portion that actually accepts the electron. Right? So all of those pigments, right, I said there's a couple hundred pigments, mostly chlorophyll, but sometimes these other pigments as well mixed in. That is the antenna complex. Right? That's like 200-300 pigments all clustered together. This is where, when that light excites that electron starts bouncing from pigment to pigment to pigment. Right? Within this antenna complex. Now if we look at the image at top, that's the image that we had been looking at. That's all those puzzle pieces kind of surrounding the center, would be the antenna complex. Another way to draw this would be down below here. We see it's drawn as a cluster. It almost kind of looks like it's drawn as a protein, but it's a cluster of pigments. All these little dots on the inside are the pigments. Right? That is the antenna complex. So this electron, it's excited, it's bouncing from pigment to pigment to pigment. Eventually, it's going to end up being captured, right? And it becomes captured at the second part of this photosystem called the reaction center. Right, so the reaction center is typically in the center of an antenna complex. And the reaction center is where that electron is captured and given to a molecule, that molecule being called the primary electron acceptor. Right? And once it's given to that molecule, it's more or less stable. It's going to stop the energy, resonance energy transfer. Right? It's going to stay within that molecule before it can leave the photosystem and do other things. All right, so we have these photosystems containing of an antenna complex, all of the pigments, and a reaction center where that electron is captured. See a lot of pens working, so I'm just going to slowly transition here. We are going to talk about two very specific photosystems when we talk about the light reactions. All right, let's do a review. So this should be an easy review, but I want you to review the concepts that we just talked about, as well as concepts that we talked about last Thursday. I'll give you a couple minutes.
Yeah. Well, they're on a damage disco ball, and it's also really and then they're like in the mosh pit where people are like throwing each other against each other. Yeah. So it's a mosh pit. Okay. Red or blue? It's blue. Yeah, blue. Because it's got rainbow. Yeah. Like whatever it absorbs, because it absorbs the other items. It's like, I want every color but red. You guys, I want. I know the first part is like, it was more like I don't like red. You guys did green, and then it was like oh now you will like it. Okay. I think I haven't taken it. I don't know. I haven't had class. I don't know. Chem is really boring, but I feel like if I jump into organic, I don't know. Yeah. Light hits an electron. That electron becomes excited. What's the one way that excitement, that excited energy can be released? Heat. All right. What's the second way? Light. And the third way? It can be transferred. Yes. And we're focusing on that resonance energy transfer bit. All right. Does red or blue light excite an electron more? Blue, yes, it has shorter wavelengths, it contains more energy, it excites that electron to an even higher energy state. All right, now we're talking about this algae, this red algae found in oceanic waters. Right? This plant appears red to our eyes. Why does it appear red? I like that answer. It absorbs every color but red, which means red is what's being reflected back in this plant. Yeah, so what wavelengths of light does this plant use to photosynthesize then? Any color that isn't red. Yes. So plants that we typically see on land outside, they're going to be green for the most part, right? They're going to utilize the light spectrum, everything besides green essentially, right? This plant looks red, right? It's using different pigments. So now it's still photosynthesizing. It's just using every color except red, because red is what's being reflected back towards us. Any questions? All right. Let's start diving into the light reactions of photosynthesis. So I mentioned that photosynthesis has two major stages. It has the light reactions, and then it has the Kelvin cycle, also called the dark reactions. Right? We're going to start by talking about the light reactions. And I think the light reactions are the harder of the two stages to conceptualize. All right, so I want to preface this by saying this is a lot of information, right? And then you're going to think, oh, we still haven't thought about the second half of photosynthesis yet. Well, the Kelvin cycle is relatively straightforward. All right, so light reactions. Big picture. That's what this slide is showing you here. The light reactions use light energy. They also use water. And what's created is oxygen, is ATP, and NADPH. And remember, NADPH is a molecule similar to NADH, like we talked about in cellular respiration. It is an energy-carrying molecule. It can carry the electrons. It holds that energy in the electrons that it carries. All right, so we're producing oxygen, which is great, right? It's actually a byproduct, but it's what we breathe. But we're producing a lot of molecules that are energy containing, whether they're an energy carrier like NADPH, or they contain energy within their bonds like ATP. All right, so if I were to show you the light reactions, right, that's a lot going on there. And we're going to break it down. We're going to simplify it. By the end of this lecture, you should be able to look at this diagram and be like, oh yeah, that's what's going on here, okay? But as we're talking about the light reactions, I really want you to focus on the movement of the electron, right? So in this image here, the electron, we first see it appearing at this first step. And as we talk about all the other steps that happen, focus on the movement of the electron through this system. All right. You are going to keep seeing this diagram coming up many times here. So if we're talking about the light reactions, we're actually going to be looking at two very specific photosystems. Right, so remember we defined a photosystem as an antenna complex with a reaction center. That's where the energy or the light is exciting the electron, right, where we're gaining that energy. That electron is bouncing from pigment to pigment to pigment before being captured in the reaction center. So in the light reactions, there are two of these photosystems. And they made it very confusing for you all to study because they're named kind of backwards, right? We have photosystem 1 and we have photosystem 2. Photosystem 1 was discovered first, which is why it's called photosystem 1. But it is not the first step in the light reactions. Photosystem 2, discovered second, is actually the first photosystem in the light reactions. Right? So it's confusing, I know. They did not make it easy for you all here. Right? But as you're thinking about this, remember that it was named according to how they discovered it, not how the process happens in the light reactions. So it's going to go photosystem 2 and then photosystem 1. And we will see that. Right? And again, I've shown you another diagram. This is essentially the same diagram as the last slide, just drawn differently, different artist's interpretation. Right? But again, we're focusing on the electrons, and they have drawn a nice red line so you can see the movement of the electron there. All right, so photosystems, they play a huge role here. Let's do a review of photosystems. So I want you to take a couple of minutes and answer these questions.
Oh, this one.
It's photosynthesis. Frogs are moist because they're like dry and free. Yeah, bullfrogs. I got some photos. And then my grandma has some I catch them all the time.
No.
Yeah, so it jumps to like all the those big boys. It doesn't matter.
You also.
Humongous fish. There's like they're not in your bed. I have never seen one like it. You could walk it like no. If you open the door and a frog jumps in, you're gonna see it. It's just like also, they don't. They're frogs. They don't want to be eaten. I feel like you should be really concerned about why you're
What would I call this cluster of pigment molecules embedded in the thylakoid membrane? I could call it the antenna complex. What else could I call it? Photosystem. Yeah, I might tend to call it a photosystem simply because I know that there is a reaction center in the middle, right? But the cluster of pigments surrounding that reaction center would all be the antenna complex. Yeah. All right, so if I have a photon striking this yellow pigment molecule, how is it going to travel to that center red dot? I like the visual there. Yeah, it might bounce around, right? It might take this path. It could take that path. It could take an entirely different path. The big picture there is that it's jumping from pigment to pigment to pigment until it reaches the center, until it reaches that reaction center. All right, what type of, what do we name this type of travel? Yes, resonance energy transfer. That is the name of this type of travel. All right, we are transferring that energy from pigment to pigment to pigment. All right, any questions? All right, we're going to start getting into the weeds of the light reactions here. All right, so like I said, photosystems were named different than how they actually appeared. So we're going to start with photosystem 2 because that is the first photosystem used in the light reactions. So if we look at this graph, photosystem 2 is the very first protein complex that we see. All right? Now in photosystem 2, this is where light is exciting the electron in those chlorophyll pigments for the first time. Right? Light is coming in, that pigment is being excited, that pigment is going to bounce from pigment to pigment, or excuse me, that electron is going to bounce from pigment to pigment to pigment until it reaches the reaction center and is captured by a molecule.
Now, when we move on to the next step, we'll see that once it's captured, it'll travel through an electron transport chain. Right, but this first step, we're just exciting the electron.
So that's all happening in that like one photosystem?
Yes, all happening in this one photosystem. Yep. So I'm actually going to flip to my next slide, which shows the exact same thing, except differently, right? Because I want you to understand the concepts and not just memorize a singular image. All right, so this is photosystem 2 as a whole. It contains an antenna complex, so we can see all the pigment molecules embedded within the complex here. Light is coming in and exciting an electron on one of the pigments. Right, again, we're following the movement of the electron here, so that electron is going to start bouncing from pigment to pigment to pigment to pigment until it reaches the center, which is the reaction center. And once it reaches the reaction center, it's going to be transferred to a molecule, right, an electron acceptor molecule, where it becomes relatively stable. All right, so electron has become excited. It's bounced around. It has finally ended up at our electron acceptor, right, making it relatively stable so it can move on to the next step. All right, but we've lost an electron. Right, so if we wanted to do this again, we would have to replace that lost electron. And that's where water comes into play. Right, so once we've lost that electron, right, given it to its electron acceptor, we need to replace that lost electron, and we do that by splitting water. Right, water is going to split, it's going to donate an electron back into the photosystem, and as a result, we're going to create some protons or some hydrogen ions, as well as some oxygen. Right, this is where we're creating that oxygen that we are breathing in. Right, this is how plants create oxygen. Through the light reactions, through the splitting of water. All right, so splitting of water has donated that electron back into the photosystem. Again, one more look at photosystem 2, because I want to make sure we understand how it's working. Right, light comes in, it excites that electron, it bounces from pigment to pigment to pigment. It ends at the reaction center, where it's given to a molecule, and that electron becomes stable. Right, that electron can now leave the photosystem and do other things. We've lost an electron, we have to replenish that electron, so we're going to do that by splitting water. Okay, and when water splits, it donates an electron, and we're left with oxygen, as well as some protons, or hydrogen ions. Right, a hydrogen ion, remember, being a proton, because it just consists of a proton. Now, creating these hydrogen ions, creating these protons, this will become important. So keep this in mind. We'll come back to this, because eventually, we're going to use those protons to make ATP. All right, so plants are pretty unique in that they can split water. There are not many organisms, not many protein complexes out there that have the ability to do this. All right, before we move on, let's look at this graph. And we're going to come back to this graph a few times. On the bottom, I'm going to put the stages of the light reactions. And along the side, I'm going to put the energy level of the electrons. I want you to tell me what is happening to the energy level of the electron in photosystem 2. And then tell me what is being produced at photosystem 2. Okay, we're getting there. Yeah, when the electron is, unless there's something we talked about last minute that just also talked about.
On right here. Yep.
Okay. I will be over there shortly.
Okay.
That's fine.
I don't think I have any.
Do you mean, do I have
Thank you very
I
much.
cannot remember this.
No, it's every time. I guess because he's worried about the backwards pass today. And I saw it. Yeah, that's fine. And it's the clear glasses. Yeah.
Okay. And remind me your name then?
Carl.
Carl. That's cool. Come back. Come back.
So much really thinking.
Yep. Very nice.
Okay, draw an arrow up or down as it hits with the photon. So when it hits, it goes up. What does produce at? What does produce at?
All right, if we're looking at the energy of this electron, which direction would I draw the arrow? Up or down? It would go up. Yes, I see some people pointing upwards. The electron is being excited, right? So it has a lot of energy in that excited state. We are increasing the energy level of our electron. All right? What is produced at photosystem 2? Oxygen. What else is being produced? Protons. Yes. We are producing oxygen. We're also producing protons, those hydrogen ions, which will become important later down the road when we talk about making ATP. All right. So we have this electron that's been excited, but it's been captured by this molecule in the reaction center and is now ready to leave photosystem 2. It's going to move on to the next step, which is an electron transport chain. There are a lot of steps here. We're going to stick to very broad basic steps here, right? So an electron transport chain, it's going to work very similar to how we saw the electron transport chain work in cellular respiration, right? That electron is going to move through a series of protein complexes, right? As the electron moves through the series of protein complexes, it's going to slowly lose its energy because that energy is being used up in order to pump the protons across the membrane. Right? We saw this happening in the electron transport chain in cellular respiration. Right? The electron moved, that movement provided enough energy to pump protons across the membrane. All right, and again, we're pumping protons across the membrane. We're starting to build up this gradient, which will be useful for making ATP. All right, I don't expect you to know all the molecules. Right? I don't expect you to know cytochrome complex. But I would expect you to know what happens to the energy level.
I don't remember, but I remember my cousin got buried all over a stranger. And they sort of escargot to Alaska.
Yep.
And that was the first of many times.
Like at a real place or like? Oh, those are my favorite.
I have had to pick a couple places though. I loved elementary school.
Electron transport chain. All the questions I ended up doing now. No, like literally just after the past like
Yeah, that's what it is.
what we create.
My grandparents have been putting basketball since I was like 5.
Really?
So now it's like back to that forever. So
And then proton gradient.
well, no, that's what I've always been playing it. It's just speeds are appropriating it. If you play this like manually, pretty complex. It's because we walked through that. We're reaching that age. I remember when I was like, when I was 8, I was like, I don't want my birthday to be 7. I'm getting too old. I'm not reaching retirement, so I kind of get the vibe of that before that. I feel like I'm gonna go missing.
Well of like optimism.
I'm gonna go missing in the woods again for sure.
All right, we're looking at our energy levels and we need to draw another arrow. What's going to happen to the energy level of our electron as it moves through the electron transport chain? If you were to show me the movement with your arms, what would you say? Up, down, going up, going down? Yes, going down, right? So you should have drawn an arrow, and the arrow is going diagonally because we're moving through the stages, and that by the time it gets to the end of the electron transport chain, right, it's going to have lost a lot of energy, right? And at the end of the electron transport chain, this electron is actually going to end up in the next stage that we will talk about, which is photosystem 1. All right, so what is happening during the electron transport chain then? What is moving? The electron. And as the electron moves, what else moves?
Protons.
Protons, yes, protons are moving across the membrane. That is key, because we're building up this proton gradient. All right. I forgot I had the answer pop up there. Any questions so far?
On the test, can I say like moves through the membrane, or do I need to say that thylakoid?
Depends on context. I prefer you say thylakoid membrane,
Okay.
but if I've already said it in the question, then membrane would probably be fine. Yeah. All right. Excuse me. Let's move on to the next photosystem. Yes, I can. All right, so we've gone through photosystem 2. All right, light has come in, it's excited this electron. That electron bounced around, reached the reaction center, where it was donated to a molecule that made it stable. The electron then left the photosystem 2, moved on to the electron transport chain, and as it moved through that chain, protons were pumped across the thylakoid membrane, starting to set up a gradient. But that electron also lost a lot of energy as it pumped those protons across. So now we're ending up at photosystem 1, right? So at this point, remember that electron has lost a lot of energy. But this electron still has more work to do. So at photosystem 1, this same electron has to be re-excited, right? It's going to gain more energy. So light is going to come in, it's going to hit photosystem 1, re-excite that same electron that used to be in photosystem 2, right? And now that it's re-excited, right, and it re-excites it the same exact way, it gets excited, it bounced from pigment to pigment to pigment, ends up in the reaction center, given away to a molecule that makes it stable, right? So same process. Once it's given away, it can move on to the next step, right? So the whole purpose of this photosystem 1 is just to re-excite that same electron. We need to give it more energy because it lost a lot of energy through the electron transport chain. Now, one thing I did not mention, I actually cut it out of the slides because I felt like it was too much detail to have to take an exam on right away. These photosystems are almost identical, but there are slight differences, right? So the name of the reaction center is different. You might see it on images. Photosystem 1, the reaction center is P700, where in photosystem 2, it's P680.
So we don't need to know that?
You don't need to know that. But if you see it, know that it's a reaction center. They're slightly different, and it references the wavelength most likely utilized by these photosystems. All right, so this electron has become re-excited, right? And now it's going to move on to the next stage, eventually to make NADPH. All right, so photosystem 1 re-excited that electron. So we see the energy level has gone up. That electron is going to go through yet another electron transport chain, right? A series of reactions that'll transport that electron from protein to protein and molecule to molecule. And ultimately, that electron is going to lose a certain amount of energy going through the electron transport chain, but it's going to end up being captured by NADP+ to make NADPH. Right? And this process from going from NADP+ to NADPH, it uses an enzyme to catalyze this reaction. That enzyme is called NADP+ reductase. Again, I don't expect you to know that name. But at this point, the electron has ended its journey, right? It's been captured by NADPH. It's not going to go any further at this point, right? NADPH is now our energy-carrying molecule and can be utilized by the cell for its energy. All right, one thing I want to point out here, and I'll point it out again in just a second. NADP+, when it turns into NADPH, it takes in a proton. It uses a proton. So that'll be important, again, when we talk about this proton gradient that we're setting up. All right, so photosystem 1 gave it a lot of energy. It's lost some energy through that second electron transport chain, and ultimately it's ended up as part of NADPH. So let's revisit our graph for the final time here. I want you to draw a couple of arrows this time about the energy levels, what happens in photosystem 1, and what happens during the electron transport chain. And tell me, why does this electron need to be re-excited?
The Nobel Prize section.
Oh,
I think there needs to be a costume competition with the theme of the Nobel Conference.
that would be hilarious.
That would be good.
Mississippi, Miami, Mississippi.
What, and I was
It might be abstract, or yeah, it'd be like,
I'm a drop of water.
the blue man, the gray.
A blue man.
Yeah,
Yeah.
there you go.
Well, you have to do it.
Not the second blue man.
No, yeah.
Great.
You're not a river, it's one of you. You're a bottle. You also have to walk with each other.
Yeah.
When it goes, so obviously when you go back up, it's getting energy back in. Is it going all the way down, or is it gonna be kind of like similar to the last electric? That would be kind of like the same arrow. Like when it gets accepted, it doesn't move wrong easily because it's stabilized.
The light again.
Okay, it
Like
was
more
still
light's
higher.
coming in and it's
Oh,
going
and that's something I haven't mentioned yet, is that when it gets open, it's actually gonna be excited even further than it ever was.
Right, because it started with more energy.
Yeah, it's vertical, yeah.
Oh, so it goes up higher?
Thank you.
It's not horizontal? Okay, but does it go up? What do you mean?
It's a little this.
Oh.
It's vertical, right? It's no longer that flat top. I don't know.
Okay, just think of it as like a the shape of the photosynthesis kind of looks like an ant.
Thylakoid membrane?
Well, not necessarily for the second electron transport chain, but it just needs more energy so it can go through the rest of the reactions it needs in order to end up at an HCH. So it's a slightly different reason. Yes, I will. Yes, I will.
Wait, so okay, I also don't like it, but
What's that?
hate them.
I never got that, so I said I'm not.
Yes. Yep. And because it's sent energy to go through the next electron transport chain. Except this chain doesn't have protons, which is why there is a reaction to get any.
Why is yours not like
That's it. Yes.
Like, wait, that was great.
You know?
My dad did almost
So you could you could have some fire. Yeah, he seems like a very
I'm going out here in the Jordan.
It's like I want you to keep an eye on the fire and think if it goes wrong.
Everybody's getting clear. I don't
Photos with the fire. As it goes to the size of like 6 foot by 6 foot. It was like,
All right, so we're talking about energy level. What happens to the energy level of the electron in the first photosystem? It goes up. Yes, it gets re-excited. It actually gets re-excited to an even higher energy state, which I did not mention. But it started with more energy initially. So when it gets excited, it's going to end up with more energy than it did in photosystem 2. All right, so that electron now has to go through another electron transport chain. What's happening to the energy?
goes down.
Goes down, yes, until it reaches NADPH. All right, so why does this electron need to be re-excited at photosystem 1? Because it lost energy, yeah. It lost energy in the first electron transport chain, and this electron still has more work to do, so it needs to gain more electrons so that it can go through the next series of reactions in order to end up at NADPH. Oh, I actually misspelled that up there. Should be NADPH, not NAPDH.
Wait, NAD what?
NADPH. I flipped the P and the D in this one. I just noticed that.
Does this whole process take like a millisecond?
It's pretty quick.
It's the speed of light.
I don't know about the speed of light, but it happens pretty quickly. Yes.
Shoot.
All right. So we have gone through this graph, and I had you do this graph specifically because this graph has a name. It's often called the Z-scheme. All right, so you'll often see this graph associated with light reactions because the light reactions is all about what is happening to that electron, what is happening to the energy in that electron. All right, so again, it's being excited from a pigment molecule in the first photosystem, which is photosystem 2. Right? It gains a lot of energy. It goes through an electron transport chain where it loses energy, but also pumps protons across the gradient. It gets re-excited at photosystem 1, gains a lot of energy, but loses a little bit of energy again through the second electron transport chain before finally being donated to NADPH. All right. And so we followed this one electron throughout the whole light reactions. If I want to do this again with another electron, where does that electron come from? Comes from water, yes. So the splitting of water will donate another electron back into this process, so we can continue making this process happen. All right. I think I have more text on the slide than I actually am talking about, because we've talked about all of this. So there's one more step to the light reactions. Remember I said we're building up this gradient of protons. And remember how I said the light reactions make ATP? Well, that is the last step, right? So throughout the light reactions, we have built up a gradient of protons. So there are more protons on the inside of a thylakoid than there are on the outside, right? And we're going to use that same protein complex we talked about in cellular respiration, ATP synthase, to now make ATP, right? And it works in the same manner. We have this proton gradient built up. The protons can only go through the membrane via ATP synthase, and the movement of those protons through ATP synthase drive phosphorylation, right? So it adds a phosphate group back to ADP to make ATP. All right, so ATP synthase, this should not be a new concept. We should know ATP synthase at this point. Now if we're thinking about how this gradient was built up, there are three ways in which it was built up. Right? First of all, right, we split water to redonate an electron in photosystem 2. When we split water, we generated some protons from the splitting of water. Right? So that increased the proton gradient inside of the thylakoid. All right, that excited electron went through that first electron transport chain, and as it moved through, it pumped some protons into the middle of our thylakoid. Right? So that increased our proton gradient inside of our thylakoids. And then our last means is when we made NADPH. Right? This time, instead of increasing the concentration inside of the thylakoid, we actually further decreased the concentration outside of the thylakoid, because we took some of these free-floating protons, combined it with NADP plus to make NADPH. Right? So we further lowered the concentration outside of the thylakoid. All of this to set up this gradient so that we can make these large amounts of ATP. All right, so at the end of the light reactions, we've made NADPH, we've made ATP, and as a byproduct from the splitting of water, we've made oxygen. Now this all happens in tandem, right? So this is a nice summary slide here, but I have this video. It takes a little bit to get started here, but this all happens together. It all happens at once. Photosystem 2 is constantly working. As soon as it loses an electron, it gains an electron and re-excites that electron to once again lose that electron. Right? And once this starts, there we go, there we have our sunlight. Our sunlight is re-exciting this electron. This electron is moving. At the same time, another electron has become excited at photosystem 2. Right? So there's multiple of these reactions happening at once. We've talked about one electron moving, but in reality, there's multiple electrons moving through at the same time. Right? And then we also saw that proton gradient is continually being built up. So we're continually making ATP. All right. Any questions about the light reactions? Right, so as you're studying, think big picture. What's going into the system? What's leaving the system? What does the path of the electron look like? And why does it move? Right? So does it move to pump protons across the gradient? Right? Does it move in that second electron transport chain to combine and make NADPH? All right, so we've talked about the light reactions. Now we have to talk about the Kelvin cycle. So the light reactions, we have made large amounts of ATP and NADPH. Right? Those energy-containing molecules are now going to be used in the Kelvin cycle. Right? So these two stages, they work hand in hand. Right? The Kelvin cycle uses the NADPH and the ATP made from the light reactions. Once it uses up those energy-containing molecules, they become NADP plus, ADP, which will then be go back into the light reactions to once again become ATP and NADPH. Right? So it's cyclic. They work hand in hand. Now the Kelvin cycle, the goal of the Kelvin cycle is not to create ATP or NADPH, but to use those molecules to create sugars. Right? We're now going to take in carbon dioxide from the air, right, and make that carbon dioxide, fix that carbon dioxide into a molecule in order to make sugar. All right. So let's dive into the Kelvin cycle. One of the biggest takeaways I want you to take away from the Kelvin cycle is that it requires large amounts of energy. Right? So to take CO2 from the atmosphere and convert that into sugar, it requires massive amounts of ATP and massive amounts of NADPH. Right? If we put some numbers to this, for every 6 molecules of carbon dioxide that are going to be converted into sugars, we have to use 18 ATP and 12 NADPH. Right? It is an energy-intensive process. That's why the Kelvin cycle really relies on the light reactions happening, because it needs such large amounts of energy. All right, so we're using a ton of energy, but we've already generated that energy in the light reactions. And what's being produced ultimately will be sugars. But the direct product of the Kelvin cycle, before it becomes a sugar, is glyceraldehyde 3-phosphate. And that molecule should ring a bell because we saw that molecule appearing in glycolysis of cellular respiration. Right? So we're actually making glyceraldehyde 3-phosphate, and then the glyceraldehyde 3-phosphates will make sugars.
Did
Yeah.
he use that for cellular respiration? Did he use that sugar?
It does use that sugar
It
through
does.
cellular respiration, yeah. Remember, plants photosynthesize, but they also go through cellular respiration. All right, so in photosynthesis, we're making those sugars, we're kind of storing that energy. So when the energy is needed, then it can be broken down via cellular respiration. So big takeaway from this slide, massive amounts of energy, lots of energy. Let's talk about the Kelvin cycle a little bit more detail, but not a ton of detail. All right, this picture has a ton of detail, and I don't expect you to know everything in this picture. We're going to break it down into big picture steps and the major players, the molecules that I would actually expect you to know something about. All right, so if you look at this image, there's a lot going on. But we can see it's broken down into 3 stages. So each kind of like triangle bit here is colored a slightly different shade, right? That indicates the different stages that are happening. So there are 3: carbon fixation, right? Reduction and carbohydrate production, and then regeneration of RUBP. Those are our 3 stages we'll talk about here. Now I think the Kelvin cycle is easier to understand because it kind of works similar to the Krebs cycle or the citric acid cycle that we've already talked about in cellular respiration. Right? Similar series of steps. So we're going to start on this first step then. Carbon fixation. All right, in this first step, this is where CO2 is entering into the picture. It's combining with a molecule, that molecule being called RUBP. Once it combines with RUBP, it becomes 3PG. Now this is where the acronyms can start to get confusing. We have G3P, and now we have 3PG, right? So it becomes 3PG, which is 3-phosphoglycerate. All right, so CO2 is coming in, it's combining with RUBP to become 3-phosphoglycerate. Now it does this with the use of an enzyme, and the enzyme's name is Rubisco. And Rubisco is a highly important enzyme. Right, and we'll talk a little bit more about Rubisco after the break. All right, so at the end point of this stage, CO2 has now combined with RUBP, and we have 3PG, right, 3-phosphoglycerate. So 3-phosphoglycerate is going to enter into the next stage of the Kelvin cycle. All right, I'm going to make sure I'm not going too fast, but that's really all I have to say about this stage. Right, we're keeping it very basic. All right, so we have 3-phosphoglycerate molecules. We actually have 12 3-phosphoglycerate molecules. We'll talk about numbers in a second here. Right. This next stage is reduction carbohydrate production. This is the stage that requires the most amount of energy. And we can see that here. Right now, there are a lot of intermediate steps that we're not going to learn about or dive into here. But ultimately, 3PG, 3-phosphoglycerate, is going to be converted into glyceraldehyde 3-phosphate, or G3P. Right? And in the process of being converted, we're using a ton of ATP, and we're using a ton of NADPH. Right, so we're turning 3PG into G3P, and those glyceraldehyde 3-phosphates will eventually go on to make sugars. Right, they can go on to make glucose. Now here's where I want us to start to pay attention to some numbers. And I don't expect you to memorize these numbers, but know the relative amounts. All right, so if we're looking at this cycle, we have 6 molecules of carbon dioxide entering into this cycle. It's going to combine with 6 molecules of RUBP in the carbon fixation stage and create 12 molecules of 3PG. Those 12 molecules of 3PG will turn into 12 molecules of G3P. This is where the numbers become important. Out of those 12 glyceraldehyde 3-phosphates that have been made, only 2 of them will actually leave this cycle and become sugars. Right, so even though we've created 12, only 2 will become a sugar. The other 10 are going to stay in this cycle and move on to the next stage. Right, so even though we're creating large amounts of glyceraldehyde 3-phosphate, only a fraction of that is being used and actually turned into sugar that can be stored by the plant and eventually used in cellular respiration. All right, those other 10, they're staying in the system, and they are moving on to our last phase, which is the regeneration of RUBP. We have to regenerate this molecule of RUBP so it can be used again. Right? We saw this in the citric acid cycle with oxaloacetate. We had to regenerate oxaloacetate so the citric acid cycle could continue. Same thing here. We have to regenerate RUBP so that it once again can be used in carbon fixation. Right now in the process, again, there's a series of steps that we aren't going to go into, but again, we're using up some energy in order to regenerate our molecule. of the Kelvin cycle. So like I said, I think the Kelvin cycle is a little bit easier to understand, but maybe it has more molecule terms, right? But big picture. CO2 is coming in. It's combining with RUBP, okay, to create an intermediate molecule, 3PG, which becomes glyceraldehyde 3-phosphate. Some of it leaves to make sugars. The rest stay in the cycle to regenerate RUBP. Alright, I slightly changed the order of the slides here. I actually have this next review question first before we talk about Rubisco. Oh, and I have this review slide, right? This is the same slide we just saw, so I'm going to just skip past it because we just talked about all of this. Right, but this is the review I want you to do. I have been known to ask something very similar to this on an exam in the past. I want you to fill in the blanks of this Kelvin cycle.
CO2.
Yeah, and it combines with
The first I don't know what it's called. Then it goes to 12.
then it switches to the other one.
Yeah, and then it goes to
And then two leaves.
G.
Oh, yeah.
G.
No, it's the.
Yeah.
And then it R's in a mix.
I don't know what it is.
Oh,
Two makes you go make sugar, and then the other 10 have to make the junction.
And then that's 6 of it. Carbon fixation.
Sorry, 22.
Do I need to know the names of the phases? Or can I
Yes,
just say
for
what they
the, I think it's 6 on the left-hand side top. And there's 6 more of these other
did? What age
molecules
do
coming
you
in.
think you're gonna Really? My grandma's, my great-grandma's dead. She was very nice. I don't know what it is. I'm gonna get the whole second camera. No, it's not. Don't lie. I'm not gonna live.
They're working on
I'm
it.
not gonna live for it. I truly do think I'll die young. Yeah. I just wanna give up. I just feel like I'm the kind of person who's dying young, you know? Like I do something stupid, or like I go skydiving. There was the biggest wildfire in
Yeah.
California.
It's a very like,
You like to go outside, because of like sugar to death. And the fire was a mountain in our house. I survived a 6.0 for.
well, if you know the diagrams, then you would be able to know scenarios, like, what happens.
See?
Oh, if this goes wrong,
I almost crushed it at my bookshelf.
still use the views with stop me.
So I was shot outside my elementary school. I've been attacked by a
Not
dog.
necessarily. If you do the acronym, that's fine.
No, he's
My
not.
only caveat is it's just so similar to PG.
I'm just
Just
trying
make
to
sure you know it's pre-PG versus PG.
There's 3 years older than me,
Yep.
and I feel my height and
But also, I don't know if this is at the end of
reduction.
So that's actually already on the.
I don't know. Yes, that's just what I can
So
remember.
G repeat leaves and makes what?
It's 2 other fires. I almost said 3 times.
or makes the sugar.
Make sugars.
Oh, the sugars?
Yeah. All right, what is the first stage of the Kelvin cycle? Carbon fixation, which means what molecule is entering into the cycle? CO2, and it's combining with what molecule? RUBP, yes, and then it moves on to the next stage, which is
Reduction and production.
reduction in carbohydrate production. Yes. I'm going to give you all the answers now. I realized I didn't do it step by step. So, right, it makes 3PG, goes into reduction and carbohydrate production, right, to make G3P, which is already on the slide, which a couple of them leave to make glucose. The rest of them stay to regenerate our RUBP. All right. I do have one more slide, but I will leave this up for a couple of minutes, or about a minute here. So on the exam, right, I know that you're just learning this information, and I'm expecting you to be tested on it on Thursday. I'm keeping that in mind when I make the exam, right? So you can expect the exam to be a lot more cellular respiration than photosynthesis. I would expect you to know much more detail on cellular respiration, fermentation, anaerobic respiration, as well as energy and enzymes, right? Photosynthesis you will still be tested on, but I'm going to keep it more broad-based.
Like this?
It could be. I haven't decided if I'm going to even go into this much detail yet. But I would know what goes in, what goes out, the major players, how everything works, all right? I'm going to be less concerned with, I think, names.
What's the 6 carbon molecule right over there by carbon fixation?
Oh, this is an intermediate. Oh.
Doesn't matter.
Oh. It is an intermediate, what we didn't talk about. Yeah. So, I won't worry about it. If you pull up the Kelvin cycle, it probably lists it. I think it's like a 110,5 phosphate group or something. I can't remember the name off the top of my head. All right. My last slide is just about Rubisco, right? That enzyme that facilitates carbon dioxide fixing with RUBP. Rubisco is a really important enzyme, right? And we think that Rubisco is the most abundant enzyme on Earth, right? Because if you think about it, think about how many plants there are. Right, now if we think about a singular plant, how many leaves does a singular plant have? All right, a singular leaf, how many mesophyll cells does a singular leaf have? Inside a mesophyll cell, how many chloroplasts are in a single mesophyll cell? Right, inside a single chloroplast, how many thylakoids are there in areas where the light reactions are happening? Right, that's a lot, right? There's a lot going on here. So Rubisco is thought to be the most abundant. We might touch upon this after break as to how Rubisco can play a role in how other plants might photosynthesize. So we talked about the most basic method of photosynthesizing, but other plants have variations on it. Maybe you've heard of a C4 plant or a CAM plant, right? Rubisco playing a role in those variations, and that's all I'm going to leave it at for now.
So we just need to know that it's abundant.
Yeah. Know it's abundant, know where it occurs. All right, so like I mentioned, there is an extra study session tomorrow, and I will be here to answer your questions. The last part of class then is I want you to finish your content check. You can probably do part 1 and part 2 before you leave class today. Part 2, you'll turn into me, and I have some study tips. Study tips, draw out these processes. That's going to be the best way to study. So content check number 4, I've also put on there like a mini exam that I did last unit as well. There's one for this unit posted on Moodle as well as the key. You can use the last, 20-ish minutes here to ask me questions, review whatever you need to review. If you want to either chalkboards to draw on, I have whiteboards that you can